respiration. All living organisms require a source of cellular energy to drive their varieties. All organisms respire by using enzyme-catalyzed reactions to release energy from energy-rich molecules, such as glucose and fatty acids, and transfer that energy to ATP. ATP is the universal energy currency of cells. In eukaryotes, aerobic respiration occurs in mitochondria. 12.1 Energy. ATP as energy, ability to do work, currency, means of exchange of goods or services. Adenosine triphosphate is a nucleotide derivative and ribose, ADE9 and 3-phosphate groups. Energy is released when ATP is hydrolysed to form ADP and a phosphate molecule. This process is catalyzed by ATP hydrolase. The inorganic phosphate can be used to phosphorylate other compounds, as a result making them more reactive. Condensation of ADP and inorganic phosphate catalyzed by ATP synthase produces ATP during photosynthesis and respiration. 12.1 to 1 Uses of energy. The ATP has the following uses: active transport across cell membranes, e.g. glucose uptake in the ileum, building macromolecules, e.g. protein from amino acids, movement by muscle contraction, endocytosis and exocytosis. ATP is a chemical energy which causes different functions to be carried, like to be carried, growth, repair, contraction, relaxation of muscles, active transport and nerve impulse, etc. Minimum amount of energy ATP required to keep the body living is 50 g or 100 cm3 of blood. Basal metabolic rate. Movement of muscles requires adenosine triphosphate, ATP, the chemical that supplies the muscles with the energy released in respiration. No ATP after death is measured. Or. Lecture notes for A-Level Biology by Muhammad Rashid Chattha. 0300 461 0457. 2. Or. ATP is a nucleotide composed of pentose sugar ribose, an organic base ADE9 and 3-phosphate groups. It is produced from the reaction of ADP and inorganic phosphate in respiration and photosynthesis. ATP is a source of energy. It is used for the process of active transport, synthesis of macromolecules from monomers, and muscle contraction. Cellular respiration is an example of a sequence of enzyme-controlled reactions involving the breakdown of complex molecules, e.g. glucose and fatty acids, to simpler molecules to release energy in the form of ATP, adenosine triphosphate. ATP is the energy storage molecule made during respiration. The stored energy is released when ATP is split to ADP, adenosine diphosphate, by losing a phosphate group. Cells use this energy for metabolic activities, e.g. synthesis of molecules and active transport. Pyrolysis of ATP to release energy. Pi is inorganic phosphate, i.e. H3PO4. When one phosphate ADP.5 is released, removal of second phosphate produces AMP and 30.5 kJ per mole energy. Removal of the last phosphate leaving adenine releases only 15.2 kJ per mole energy. A metabolic pathway is a sequence of enzyme-controlled reactions, and molecules involved are called metabolites. Enzymes are important because they are biological catalysts, highly specific, and their production is controlled by the nucleus. In anabolic reactions, complex molecules are built up from simpler ones, and in catabolic reactions, complex molecules are broken down, e.g. respiration. Cell respiration is the release of energy from organic molecules. The energy released is transferred to ATP, a compound that can provide the cell with energy in small, instant, controllable amounts. Cell respiration consists of many reactions. Each reaction is controlled by a different enzyme. Most of the reactions occur inside the mitochondrion. Lecture notes for A-Level Biology by Muhammad Rashid Chattha. 0300 461 0457. 3. enzyme-controlled steps by which an organic molecule, usually glucose, is broken down so that its chemical potential energy can be used to make ATP. In aerobic respiration, the sequence involves four main stages: glycolysis, the link reaction, Krebs cycle, and oxidative phosphorylation. The acetyl enzyme A enters the Krebs cycle in the mitochondrial matrix and donates the acetyl unit to acetate, for C, to make citrate, 6C. The Krebs cycle decarboxylates and dehydrogenates citrate to oxaloacetate in a series of small steps. The oxaloacetate can then react with another acetyl enzyme A from the link reaction. 12.1-2 Features of ATP as universal currency. ATP is used as the universal energy currency. ATP can be synthesized from ADP and phosphate using energy, and hydrolyzed to ADP and phosphate to release energy. 12.1-3 ATP is synthesized by transfer of phosphate in substrate-linked reaction and chemiosmosis in membranes of mitochondria and chloroplast. ATP is synthesized by substrate level, when ATP is made from ADP and a phosphate group which is transferred from a highly reactive intermediate. An example would be during glycolysis. ADP joins with the inorganic phosphates transferred from triose bisphosphate molecules forming 4 ATP per glucose molecules. This also happens in the Krebs cycle. Chemiosmosis, the movement of protons across a membrane which synthesizes ATP. Protons diffuse down a concentration gradient through a partially permeable membrane. As protons flow down, energy is released. The energy is used in the attachment of ADP to an inorganic phosphate. 12.1-4 Relative energy values of carbohydrates, lipids, and proteins as respiratory substrates. A single factor for each of the rates, protein, carbohydrate, regardless of the food in which it is found. The energy values are 17 kJ/g, 4.0 kilocalories per g, for carbohydrates. 37 kJ/g, 9.0 kilocalories per g, for fat. 17 kJ/g, 4.0 kilocalories per g, for protein. 12.1-5 Using different respiratory quotient, RQ. A respiratory substrate is the organic substance which forms the starting point of respiration. So far we have only looked at the respiratory pathway involving glucose as a respiratory substrate is to calculate its respiratory quotient, RQ. RQ value of carbohydrates is equal to 1.0, amino acids, proteins, equals 0.9, fats, triglycerides, equals 0.7. A respiratory quotient is calculated by Lecture Notes for A-Level Biology by Muhammad Rashid Chattha. 0300 461 0457. 4. If the value is 1.0 or below, it indicates that aerobic respiration is occurring, and 1.0 or above indicates anaerobic respiration. The value also indicates the substrate being used. RQ value of different substrates. Carbohydrate equals protein equals 0.9 equals 0.7. Higher respiratory quotients are attained in animals converting carbohydrate to fat in preparation for hibernation. 12.1-6 Calculation of RQ values from equations of respiration. Using different respiratory substrates. 12.1-7 Investigations using simple respirometer to determine the RQ of germinating seeds or small invertebrates. Respirometers are used to measure and investigate the rate of oxygen consumption during respiration in organisms. They can also be used to calculate respiratory quotients. The experiments usually involve organisms such as seeds or vertebrates. Lecture Notes for A-Level Biology by Muhammad Rashid Chattha. 0300 461 0457. 5. 12.2 Respiration. Respiration in all living cells. It is a biochemical process in which organic substances are used as fuel. They are broken down in a series of stages and the chemical potential energy they contain is transferred to another molecule, ATP. This provides an immediate source of energy for various forms of biological work such as movement, the synthesis of large organic molecules and active transport. You may be familiar with the simple equation sometimes used to summarize the process of respiration. C6H12O6 plus 6O26CO2 plus 6H2O plus energy. 12.2 to 1/4 stages of aerobic respiration occurs in eukaryotic cells. The overall process of glucose down. Stages 2, 3, and 4 can be thought of as the aerobic, oxygen-requiring reactions and take place in the mitochondria. Stage site within several process number of ATP molecules produced. 1. Glycolysis cytosol, cytoplasm glucose is split into 2 molecules of pyruvate. 2 per glucose. 2. Pyruvate matrix, inner fluid, of mitochondria pyruvate is converted into acetyl-CoA none. 3. Krebs cycle matrix, inner fluid, of mitochondria acetyl-CoA drives a cycle of reactions which produces hydrogen. 2 per turn, so 4 per glucose. 4. Electron transport chain inner membrane of mitochondria hydrogen drives a series of redox reactions, which release enough energy to make ATP. Up to 32 per glucose. A respiration of oxygen and results in the production of 38 ATP molecules, water and carbon dioxide. There are 3 main processes. Glycolysis occurs in cytoplasm. 2. Link reaction in mitochondrial matrix. 3. Krebs cycle in mitochondrial matrix. 4. Electron transport chain on the inner membrane of mitochondria. Complete erosion of glucose 4 stages: glycolysis, pyruvate oxidation, the reactions of the Krebs cycle and the reactions of the electron transport chain. 1. Glycolysis, glucose is broken down into 2 molecules of pyruvate. Only 2 molecules of ATP are made. This happens in the cytosol of cells. Lecture notes for A-Level Biology by Muhammad Rashid Chattha. 0300 for 610457. 6. 2. Pyruvate oxidation, the link reaction in the presence of oxygen. Pyruvate enters the mitochondrion and is converted into acetyl coenzyme A. 3. The Krebs cycle, a series of reactions, fueled by acetyl coenzyme A, which produces only 2 molecules of ATP but many electrons and hydrogen ions. 4. Oxidative phosphorylation. For stages of respiration. Glycolysis takes place in the cytoplasm of the cell and involves the oxidation of glucose to the end product, pyruvate. Initially, the glucose is phosphorylated using 2 molecules of ATP to make it more unstable, so that it readily breaks down into 2 molecules of glycerate 3-phosphate. A net gain of 2 ATP molecules and reduced NAD are produced. Pyruvate enters the matrix of a mitochondrion and undergoes a link reaction. It is converted to acetyl, a 2-carbon molecule, which combines with coenzyme carbon molecule, oxaloacetate, to form a 6-carbon molecule, citrate. Many intermediate redox reactions occur, resulting in the release of carbon dioxide, hydrogen ions, and energy in the form of heat and ATP. The hydrogen ions are removed by enzymes called dehydrogenase and reduce the coenzymes NAD and FAD. The process of oxidative phosphorylation takes place on the cristae of a mitochondrion. This is called the electron transport chain, where electrons from the reduced coenzymes pass to a chain of electron carriers involving redox reactions. The energy released is used to form ATP and the final hydrogen acceptor is oxygen, which forms water. Anaerobic respiration occurs in the absence of oxygen and therefore only glycolysis can take place. In muscles, the product is lactic acid, which is toxic and causes cramp. In yeast, the products are ethanol, carbon dioxide, and NAD, and it is used in the brewing industry. Overall, only 2 ATP molecules are produced, compared to 38 ATP molecules in aerobic respiration. i.e. aerobic respiration is more efficient. 12.2 Glycolysis. Glucose is split into two pyruvate molecules. Glycolysis is the first step in the biochemical pathway of respiration. A molecule of glucose is broken down into two pyruvate groups, each of which contains 3 carbon atoms. Glucose contains a lot of chemical potential energy and is not a very reactive substance. In order to release this energy, we need to use some energy from ATP. In the first stage of glycolysis, a molecule of glucose is converted into 2 molecules of triose phosphate. This requires 2 molecules of ATP. Triose phosphate is then converted to pyruvate. This process produces 4 molecules of ATP, 2 for each triose phosphate. The conversion of triose phosphate to pyruvate is an oxidation and involves the transfer of hydrogen to a carrier molecule. This carrier molecule or coenzyme is known as NAD, nicotinamide adenine dinucleotide. Adding hydrogen reduces NAD to reduced NAD. Coenzymes like NAD, FAD, flavine adenine dinucleotide, and coenzyme A are involved in respiration. Coenzyme is a molecule required for an enzyme to be able to catalyze a reaction. NAD and FAD are reduced during respiration. The term reduced means to add hydrogen, so reduced NAD hydrogen added to it. Without the presence NAD or FAD except the hydrogen, the dehydrogenase enzymes involved in respiration would not be able to remove hydrogen from their substrates. Summarizing glycolysis molecule, we get 2 pyruvate groups, a net gain of 2 molecules of ATP and 2 molecules of reduced NAD. Lecture notes for A-Level Biology by Muhammad Rashid Chattha. 0300 for 610457. 7. 12.2-3 The link reaction, pyruvate oxidation, is oxidized into acetate. Pyruvate still contains a lot of chemical potential energy. When oxygen is available, this energy can be released in a cycle of reactions known as the Krebs cycle. This link reaction is a term used to describe the reaction linking glycolysis and the Krebs cycle. 12.2-4 Acetyl coenzyme A pyruvate combines with coenzyme A to produce acetyl coenzyme A. This involves the loss of a molecule of carbon dioxide. It is also an oxidation NAD. Lecture notes for A-Level Biology by Muhammad Rashid Chattha. 0300 for 610457. 8. 12.2-5 The Krebs cycle. Electrons are stripped off the acetate. The Krebs cycle is a cycle of reaction which involves a number of intermediate steps. We will only concern ourselves here with its main features. Acetyl coenzyme A produced in the link reaction is fed into the cycle 6-carbon compound, citrate. In series of reactions, the citrate is converted back to oxaloacetate. This involves the production of 2 molecules of carbon dioxide, which is given off as a waste gas. For each complete turn of the Krebs cycle, 1 molecule of ATP is produced. The reactions which form the Krebs cycle again involve oxidation. The hydrogens which are given off are used to reduce coenzymes. Each turn of the cycle produces 3 molecules of reduced NAD and 1 molecule of another reduced enzyme, reduced FAD. The most important function of the Krebs cycle in respiration is the production of reduced coenzymes. These are passed to the electron transport chain where the chemical potential energy these molecules contain is used to produce ATP. Lecture notes for A-Level Biology by Muhammad Rashid Chattha. 0300 for 610457. 9. 2-6 Decarboxylation, dehydrogenation. Dehydrogenation provides hydrogen atoms, which are accepted by the carriers NAD and FAD. These pass to the inner membrane of the mitochondrial envelope, where the hydrogen atoms are split into protons and electrons. Adenosine triphosphate is a nucleotide and consists of 3 parts. It contains an organic base, adenine, a pentose sugar, ribose, and 3 phosphate groups. It is formed in the processes of respiration and photosynthesis from adenosine diphosphate, ADP, an inorganic phosphate, pi. When a molecule of ADP is hydrolysed, an exergonic reaction occurs. This releases about 33 kJ/mol of energy. A metabolic pathway series of catalyzed by enzymes. An anabolic reaction occurs when X molecules are built up from simpler ones. 12.2 to 7 The electron transport chain. The energy in the electron is used to make ATP. The reduced NAD and reduced FAD produced as a result of oxidation now pass to chains of molecules situated on the internal membranes in the mitochondria. These are the electron transport chains. The hydrogen is removed from the 2 reduced coenzymes and is split into hydrogen ions, 8, and electrons. The electrons pass from one molecule to the next along the electron transport chain. At each transfer, a small amount of energy is released. This is used to pump the hydrogen ions out through the inner mitochondrial membrane in the space between the inner and Lecture Notes for A-Level Biology by Muhammad Rashid Chattha. 0300 461 0457. 10. The outer membrane. When the hydrogen ions return into the matrix of the mitochondrion, they release their potential energy. This is used to produce ATP. The last molecule in the chain is oxygen. Oxygen combines with hydrogen ions and electrons to produce water. Lecture Notes for A-Level Biology by Muhammad Rashid Chattha. 0300 461 0457. 11. 12.2 to 8 Synthesis of ATP by phosphorylation, oxidative phosphorylation. ATP is made by adding a phosphate group to a molecule of ADP. This process is called phosphorylation and it is carried out in 2 different ways. Substrate-level phosphorylation is the process in which a phosphate group from a substrate molecule, a molecule other than ATP, AEP or AMP, is transferred to a molecule of ADP, giving a new molecule of ATP. This form of phosphorylation occurs in glycolysis and in the Krebs cycle. In oxidative phosphorylation, ATP is synthesized using free phosphate groups. The energy required is obtained from a series of redox reactions in the electron transport chain. So when you see oxidative phosphorylation, think electron transport chain. Electron transport chain, a series of redox reactions, fueled by the electrons and protons from the preceding 3 processes, as supplied by the coenzymes NAD and FAD. Electron transport releases enough energy to synthesize most of the ATP produced by aerobic respiration. Chemiosmosis. In biology, chemiosmosis refers to the process of moving ion, e.g. protons, to the other side of a biological membrane, and as a result, an electrochemical gradient is generated. This can then be used to drive ATP synthesis. Most food chemicals, carbohydrates, lipids, and proteins, can be respired to provide energy. Most organisms respire aerobically, in the presence of oxygen. The substrate, e.g. glucose, is broken down completely to produce carbon dioxide, water, and a lot of ATP. Lecture Notes for A-Level Biology by Muhammad Rashid Chattha. 0300 461 0457. 12. And some tissues respire anaerobically. In the absence of oxygen, substrates are only partially broken down, leading to waste products such as lactate, in animals and bacteria, or ethanol and carbon dioxide, in plants and yeast. Organisms require energy to stay alive. The energy used is normally the chemical potential energy of organic molecules, especially carbohydrates and lipids. The uses of the energy include anabolic reactions, active transport, and movement. 12.2 to 9 phosphorylation. In oxidative phosphorylation, electrons are passed along a series of carriers. Some of the energy released as this happens is to move protons from the mitochondrial matrix to the intermembrane space. This sets up a gradient of protons across the inner membrane of the mitochondrial envelope. Protons pass back into the matrix, moving down their concentration gradient by facilitated diffusion through protein channels in the inner membrane. An enzyme, ATP synthase, is associated with each of the proton channels. ATP synthase uses the energy of the proton gradient to phosphorylate ADP and ATP. At the end of the carrier chain, electrons and protons are recombined and reduce oxygen to water. In the absence of oxygen, ethanol fermentation and lactate fermentation, a sum of ATP is made through glycolysis, then removing hydrogen and produce ethanol or lactate. Glycolysis is the first stage of respiration and involves the breakdown of glucose in the cytoplasm to produce pyruvate. Glucose is phosphorylated using phosphate ions from two ATP molecules. This increases reactivity of the glucose-forming fructose 1-6 bisphosphate. Unstable fructose 1-6 bisphosphate breaks to two molecules of glyceraldehyde 3-phosphate. Each glyceride 3 and a pair of H+ ions to NAD. NAD is reduced, producing two NADH2 molecules for ATP molecules and pyruvate. From one molecule of glucose, glycolysis generates two molecules of pyruvate, two NADH2, reduced NAD, and two ATP overall. Exercise places great metabolic demands on the body. Homeostatic mechanisms must cope with a much greater oxygen demand as well as a buildup of carbon. Notes for A Level Biology by Muhammad Rashid Chattha. 0300 461 0457. 13. Dioxide. In the long term, there may be a significant loss of water and salts, electrolytes, through seating. There are three sources of ATP. These comprise three energy systems. The most immediate is the ATP-ACP, or lactic anaerobic system. ATP already present in the muscles is backed up by creatine phosphate, which is used to make more ATP instantly. The second system that kicks into action is the glycolytic or lactic anaerobic system. ATP is supplied by glycolysis, and this system results in a buildup of lactate ions. The third is the aerobic system, in which energy comes from the complete oxidation of glucose. The duration of an event determines the type of energy system used, up to 10 seconds, the ATP-ACP system, from 1 to 60 seconds, the glycolytic system, and more than 60 seconds, the aerobic system. In the short term, the body responds to exercise by maintaining homeostasis. There is an increase in heartbeat ventilation rate, coupled with vasodilation and sweating. During exercise, we can build up an oxygen debt. This is off after exercise has finished, or during rest periods within the activity. This is why pulse and ventilation rate remain higher than normal even when activity has stopped. Extra oxygen is needed to replace the ATP-ACP stores and to oxidize accumulated lactate. The long-term responses of the body to exercise are generally those that improve the body's fitness. These include strengthening of the cardiovascular system, heart and blood vessels, and making the lungs and muscles more efficient. The overall result is a body that is better able to cope with the chosen activity. CM plants save water by opening their stomata at night, storing carbon dioxide in an organic acid before releasing it for photosynthesis in the daytime. C4 plants avoid photorespiration, allowing them to make use of all the available carbon dioxide and therefore to grow faster. The 12th of February, 10 anaerobic respiration. Sometimes there is not enough oxygen for an organism to respire using the pathway we have just described. Under these conditions, it has to produce ATP by respiring anaerobically. The only stage in the anaerobic pathway which produces ATP is glycolysis, so it is not as efficient as aerobic respiration. The chemical potential energy from a single molecule of glucose can be used to produce 38 molecules of ATP in Anaerobic respiration. It can only produce 2 molecules of ATP in anaerobic respiration. During glycolysis, NAD is reduced. Reduced NAD is normally converted back to NAD when nitrogen is transferred in the electron transport chain. This will only happen when oxygen is present. Obviously, if all the NAD in a cell was converted to reduced NAD, the process of respiration would stop. In anaerobic respiration in animals, pyruvate is converted to lactate. In plants and microorganisms such as yeast, involves the conversion of reduced NAD to NADH and allows glycolysis to continue. Lecture notes for a level biology by Muhammad Rashid Chatter. 0300 461 0457. 14. It is really an equation for the complete oxidation of glucose and is misleading in a number of ways. It shows the fuel as glucose. In many living cells, although the main fuel is glucose acids, and amino acids are also respiratory substrates and can be used for respiration. The equation also shows that oxygen is required. Respiration can, however, take place anaerobically, that is, without the presence of free oxygen. Finally, the equation fails to show that respiration involves a number of reactions in which the respiratory substrate is broken down, in a series of steps, releasing a small amount of energy each time. We will look in more detail at glucose breakdown. It involves the 4 steps given below: glycolysis, link reaction, Krebs cycle, and electron transport chain, ETP. The 12th of February, aerobic and anaerobic respiration. Aerobic respiration requires oxygen and it involves main stages called glycolysis, the Krebs cycle, and the electron transport chain. Glycolysis involves the conversion of glucose to pyruvate with a net gain of ATP and reduced NAD. Initially, the glucose undergoes a phosphorylation reaction to make it more unstable, and break down into 2 molecules each containing 3 carbon atoms, called glycerate 3-phosphate. The Krebs cycle occurs in the matrix of a mitochondrion. Pyruvate is oxidized and results in the production of carbon dioxide, ADH+, H+, and ATP. The oxidative phosphorylation occurs on the electron transport chain on the cristae of the mitochondrion. This results in the generation of ATP. The product of anaerobic respiration in muscles is called lactic acid, and in yeast, ethanol and carbon dioxide are produced. Anaerobic respiration occurs in the absence of oxygen and produces very less amount of energy, i.e. 2 ATPs only. Whereas in the case of aerobic respiration, 36 ATPs are produced. Redox indicators are used to demonstrate oxidation reactions. Methylene blue changes from blue to colorless when hydrogen ions are accepted. If a respiratory quotient of 1 is obtained, the substrate is usually a carbohydrate. The value can be calculated by the volume of carbon dioxide produced divided by the volume of oxygen absorbed. 12th of February, 12 rice plant adaptations, parenchyma tissue. Rice plant has it survive in anaerobic conditions. It survives submerged in water or in dry conditions. Rice provides staple food to more than half of the world population. Rice is a partial hydrophyte plant grown in paddy fields having plenty of water. Plants show adaptations to different environments. The adaptations to swamp conditions by rice include extensive parenchyma tissue, with continuous, interconnecting air spaces through stems, leaves, and roots, and physiological tolerance of ethanol by root cells. The cells of the embryo are tolerant to the high concentrations of ethanol that build up as a result of anaerobic respiration. Lecture by Muhammad Rashid Chatter. 0300 461 0457. 15. The stems and root interfaces between the cells which allow oxygen to diffuse, not submerged, and allow the cells and tissue to respire aerobically. This type of tissue is called parenchyma. If the root salt of oxygen, they respire anaerobically and tolerate the build of ethanol. They also produce an abundance of the enzyme alcohol dehydrogenase, which breaks the ethanol down. Some varieties of rice have higher rate of anaerobic respiration to increase the rate of ATP production. The 12th of February, 13 oxidation reduction reaction. Redox indicators are used to show when an oxidation reaction has taken place, i.e., the addition of oxygen, the removal of hydrogen ions or electrons. Methylene blue acts as an artificial hydrogen acceptor which is blue, oxidized form, and turns colorless, reduced form. Tetrazolium chloride changes from colorless, oxidized form, to pink, reduced form. These can be used as useful indicators of the speed of the action of enzymes. During the experiment, oxygen is prevented from entering to stop the oxidation of the methylene blue. A calorimeter can be used to measure the percentage of light transmitted. Redox indicators can be used to measure the rate of respiration in yeast, and therefore to investigate the effect of factors such as temperature or substrate on concentration on the rate of respiration. Of February, 14 respirometer. The energy values of respiratory substrates depend on the number of hydrogen atoms per molecule. Lipids have a higher energy density than carbohydrates or proteins. The respiratory quotient, RQ, is the ratio of the volume substrate being respired. Carbohydrate has an RQ of 1.0, 0.17, 0.9. Rate of oxygen uptake, and hence RQ, can be measured using a respirometer. The rate of respiration can be measured as a respiratory quotient, the ratio of CO2 evolved to O2 absorbed. It can be calculated from measurements made using a respirometer. Keywords: acetyl-coenzyme A, adenosine triphosphate, ATP, erenchyma, aerobic respiration, anabolic reactions, anaerobic respiration, ATP synthase, chemiosmosis, citrate, coenzyme, coenzyme A, Christi, DCIP, decarboxylation, dehydrogenation, electron acceptor, electron carriers, electron transport chain, FAD, fructose 1,6-biphosphate, glucose phosphate, glycolysis, inner mitochondrial membrane, intermembrane space, oxygen debt, redox indicator. Respiratory quotient, respiratory substrate, respirometer, substrate level phosphorylation, triose phosphate. Lecture notes for Level Biology by Muhammad Rashid Chattha. 0300 461 0457. 16. Chapter Glossary. Acetyl coenzyme A is a 2-carbon molecule formed in the link reaction when an acetyl group is bound by coenzyme A. It is oxidized in the Krebs cycle. Adenosine triphosphate, ATP, is the universal energy carrier found in all living cells. Erenchyma is a spongy tissue with air spaces that allow gas exchange between the shoots and the roots of some plants, e.g. rice. Aerobic respiration is a form of cellular respiration that takes place in the presence of oxygen and produces carbon dioxide, water, and ATP. Overall, C6H12O6 plus 6026CO2 plus 6H2O anabolic reactions are those reactions in which smaller molecules react to form larger molecules. This requires energy input. Anaerobic respiration is a form of cellular respiration that takes place in the absence of oxygen. It produces less ATP than aerobic respiration. ATP synthase is an enzyme found embedded in cellular membranes that phosphorylates ADP to form ATP as protons flow through it. Synthase is the synthesis of ATP through the movement of protons down there. Concentration gradient across a semi-permeable membrane, catalyzed by ATP synthase. Citrate is a 6-molecule formed in the first stage of the Krebs cycle from the reaction of acetyl coenzyme A and oxaloacetate. Coenzyme is a molecule that helps enzymes carry out their function, e.g. NAD, FAD, coenzyme A. Coenzyme is a coenzyme that binds an acetyl group to form acetyl coenzyme A during the link reaction of aerobic respiration. Cristae are folds of the inner mitochondrial membrane that provide a large surface area for oxidative phosphorylation. Decarboxylation is the removal of a carbon dioxide molecule. Dehydrogenation is the removal of a hydrogen atom. Oxygen acts as the final electron acceptor in the electron transfer chain, 1/2 O2 plus 2E, plus 2H plus H2O. Electron carriers are protein molecules that accept and release electrons, e.g. NAD. Electron transport chain is a series of electron carrier proteins that transfer electrons in a chain reduction reactions. FAD is a keenzyme that becomes reduced when it takes up hydrogen atoms during the Krebs cycle, forming reduced FAD. Fructose 1,6-bisphosphate is a 6-carbon molecule formed by the relation of glucose during glycolysis. It splits into 2 molecules of triose phosphate. Glucose is a 6-carbon monosaccharide, which is an important respiratory substrate. Glycolysis is an anaerobic process that takes place in the cytosol of the cell and breaks down glucose into 2 molecules of pyruvate. 2 molecules of ATP and 2 molecules of reduced NAD are also formed. Inner mitochondrial membrane is the mitochondrial membrane that separates the matrix from intermembrane space. It is the site of the electron transport chain. Intermembrane space is the small space between the inner and outer mitochondrial membranes. The electron transport chain results in a high proton concentration here. Krebs cycle is a series of oxidation-reduction reactions in the matrix of the mitochondria in which acetyl coenzyme A is oxidized, generating reduced NAD, reduced FAD, ATP, and carbon dioxide. Lecture notes for A-Level Biology by Muhammad Rashid Chattha. 0300 for 610457. 17. Lactate dehydrogenase is an enzyme that catalyzes the conversion of pyruvate to lactate. Reaction is the first stage of aerobic respiration, also known as oxidative decarboxylation. It takes place in the mitochondrial matrix and converts pyruvate into acetyl coenzyme A and carbon dioxide. Reduced NAD is also formed. Overall, pyruvate plus NAD plus acetyl-CoA plus reduced NAD plus CO2 methylene blue is an artificial hydrogen acceptor that changes color from blue to colorless when reduced. Mitochondrial matrix is the fluid-filled base within the inner membrane of the mitochondria which contains mitochondrial DNA and enzymes required for aerobic respiration. Mitochondrion is an organelle found in eukaryotic cells that is the site of aerobic respiration. NAD is a keenzyme that becomes reduced when it takes hydrogen atoms during aerobic respiration, forming reduced NAD. Outer mitochondrial membrane is the membrane segregating the contents of the mitochondrion from the rest of the cell. It creates optimal conditions for aerobic respiration. Oxaloacetate is a 4-carbon molecule coenzyme A to produce 6-carbon citrate in the cycle. It is eventually regenerated, allowing the cycle to continue. Oxidative phosphorylation is the synthesis of ATP from reduced coenzymes and oxygen in the electron transport chain of aerobic respiration. Oxygen debt is the amount of additional oxygen required after a burst of anaerobic respiration to restore the body to a resting state. Pyruvate is a 3-carbon molecule produced in glycolysis. In the link reaction of aerobic respiration, it is oxidized to acetate. During fermentation, it is converted to lactate, animals, or ethanol and carbon dioxide, plants and microorganisms. Redox indicator is a substance which changes color when it gains or loses electrons. Respiratory quotient is the ratio of carbon dioxide produced to oxygen consumed during respiration. Calculated respiratory substrate is an organic molecule that can be broken down via the respiratory pathways to produce ATP. Respirometer is a device used to determine respiration rate in living organisms by measuring the change in volume of oxygen or carbon dioxide. Substrate-level phosphorylation is the synthesis of ATP by the transfer of a phosphate group from a phosphorylated intermediate to ADP. Triose phosphate is a 3-carbon compound formed in glycolysis. Lecture notes for 11 Biology by Muhammad Rashid Chattha. 0300 461 0457. 18. 13. Photosynthesis. Photosynthesis is the energy transfer process, i.e., the basis of nearly all life on Earth. It provides energy directly or indirectly to all the organisms in most food chains. In eukaryotes, the process occurs within chloroplasts and leaf